Welcome to our educational webinar titled Recreating Disease with Kidney Organoids and CRISPR. My name is Andrew Gaffney and I'm the Product Manager for Pluripotent Stem Cell Biology at STEMCELL Technologies, and I'll be your moderator for this webinar. It is now my pleasure to introduce you to our speaker today. Dr. Benjamin Freedman is an Assistant Professor at the University of Washington, Department of Medicine, Division of Nephrology. He is also a member of the Kidney Research Institute and Institute for Stem Cell and Regenerative Medicine. Dr. Freedman first became fascinated with stem cells as an undergraduate at the University of Pennsylvania, where he performed laboratory studies into mammalian epimorphic regeneration after injury. He went on to earn his doctorate in molecular and cell biology from the University of California at Berkeley, where he learned to reconstitute the process of nuclear remodeling in cytoplasmic extracts from frog eggs. As a postdoctoral fellow in the renal division at Brigham and Women's Hospital at Harvard Medical School, Dr. Freedman was responsible for innovative techniques, including differentiation of human pluripotent stem cells into kidney organoids and using CRISPR-Cas9 genome editing to model kidney disease. He currently leads a research group at the University of Washington in Seattle devoted to the reconstitution of regeneration disease mechanisms in organoids to guide therapy development. Let me now hand you over to Benjamin to begin the presentation.
Thanks very much. And thank you to the kind folks at STEMCELL for inviting me to talk today. And it really gives me great pleasure to discuss our work, recreating disease with kidney organoids and CRISPR. I thought I'd start with this quote from Dr. Richard Feynman, who was not a biologist, but rather a physicist in the 20th century. So Dr. Feynman taught us that what I cannot create, I do not understand. And I think this is a principle that holds true in biology as much as it does in physics. And the concept is that our ability to engineer a phenomenon with our own hands is really a metric of how well we understand the underlying principles of that phenomenon. So in biology, we can actually do this in a real physical way, using a variety of different models. And these include in vitro models, which are shown on the left here, which are generally cells. And these can be very high throughput. They can be mechanistic. We can take cells from humans. But on the downside, these in vitro models tend to be very simplistic compared to what's actually going on in an actual living organism. We also have in vivo models, such as the laboratory mouse, and these are more complex and more physiological compared to cells in a dish, but they have the disadvantage of being low throughput, and they tend to be more descriptive in the experimental types of approaches, and obviously we're very limited in terms of what we can do in humans in vivo. So a new emerging intermediate model is the organoid. And on the left here, you have some pictures of various organoids that have been produced. This is actually a cover from Nature Methods last year, where organoids were picked as the method of the year. And here are actually what these look like after they've been immunostained for various different types of organs. And you can see that there are a variety of different patterns that emerge in these organoids. And it's really that complex patterning that is the hallmark of the organoid. So what is an organoid? And my definition of an organoid is the following. I think an organoid is a multicellular unit in vitro that resembles a tissue or organ of the body structurally and functionally. And in general, this would involve complex shapes, and it also involves having different types of cells within the structure. So our particular organ of interest is the kidney, and the kidney is highly complex. And this is a picture of a kidney here. At the meta level, you have a blood supply coming in, a blood supply coming out, and then a filtrate draining into the ureter, which is the urine. At the more micro level, each kidney actually is composed of about 1 million different little tiny subunits called nephrons. And this is an image of a nephron here. And the way the nephron works is that blood flows in and it enters a capillary nest. This is known as the glomerulus, and it's perfused through a layer of epithelial cells here, which are called podocytes. These are filtering cells. They prevent red blood cells and most of the protein from coming in, and the perfusate enters a long, convoluted tubular structure, which makes up the bulk of the nephron. And this tubular structure is intimately opposed to capillaries, so that there can be exchange of nutrients and molecules from the blood into the tubules and vice versa. And ultimately, what ends up coming out of the nephron are only the waste products and the undesirable or unneeded elements in our blood, whereas almost everything that we want or that we want to keep gets taken back up into the body. So this is a very complicated little structure. And we've recently been able to generate organoids that simulate aspects of this nephron, and this is shown here. This is an issue of Stem Cells that just came out last year, where we had a paper, and on the cover we show these filtering cells, which are shown in red here, called podocytes, and these green nuclei are the tubular cells. And you can see that the tubular staining pattern is reciprocal to the podocyte staining pattern. And this structure sort of rudimentarily resembles what's existing in the kidney nephron. So in today's talk, it's going to be broken up into 3 parts. First, I'm going to discuss briefly about how we've generated human kidney organoids for the first time. And then we're going to discuss how we've used CRISPR gene editing to recreate human disease in these organoids. And finally, we'll break into some future directions and conclusions, and hopefully some fun questions from the audience. So first, how did we make the kidney organoid? So because the kidneys only form during embryonic development, we knew that we would have to start with stem cells. And in particular, we have to go all the way back to the start to pluripotent stem cells. So that the approach that we've taken is somewhat similar to the general approach for regenerating many different organs. And this is, we start with a doctor and a patient, and this is, you'll have to forgive my sort of antiquated picture here of this interaction. But the doctor procures adult cells from the patient. These are not stem cells. They can come from any quickly growing type of cell, skin, blood, anything that can grow. And then by introducing into these adult cells stem cell genes and growth factors, they can be reprogrammed back to an embryonic stem cell-like state. And these are called induced pluripotent stem cells, or iPS cells for short. And I think that's a great example, by the way, of how understanding a phenomenon allows us to recreate it, because we've actually taken an adult cell and taken it back to a stage that didn't exist before. And now, with these reprogrammed cells, what's really new here is that we've been able to generate kidney organoids from these structures, and we can now use these organoids in vitro for disease modeling applications. And in the long term, we can think about growing not just organoids, but actually whole organs that could be then self-transplanted back into the patient. And these would potentially be 100% immunocompatible with that patient. And that's very important for kidney disease, because kidneys are the most commonly transplanted organ, but there is a great shortage of available kidneys for transplant. And even in the patients who are lucky enough to receive a kidney transplant, they'll need to take immunosuppressive medications because those transplants are coming from another human being. And those immunosuppressive medications, over the course of the life of the graft, can be very difficult and can cause harmful side effects. So we're proposing an alternative sort of therapy here in the long term, which would involve regenerating a person's kidney from their own body, and that could be done on demand and would not require immunosuppression. Obviously, that's a long way in the future. We're not close to being there yet, but we are very close to using these for disease experiments in drug profiling, as I will discuss. So I wanted to introduce our protocol for differentiating iPS cells into kidney organoids. And this is a protocol that we described in 2015 in a Nature Communications paper shown here. And it's really relatively simple, I think, compared to some of the other protocols out there that emerged around the same time. And all we have to do to make kidney organoids in this protocol is start with dissociated iPS cells. These are undifferentiated. We typically grow them in M-teaser. And then we sandwich them in a thin layer of Matrigel. And we treat them subsequently. They form these spheroid structures, which are still undifferentiated. And we treat them subsequently with a GSK3 beta inhibitor called Chiron 99021. And we use a high dose of this compound. And what this does is it induces the differentiation of the pluripotent stem cells into what are called nephron progenitor cells, or NPC. And these are cells, these are stem cells that arise during kidney development that are responsible for the growth and differentiation of the nephron structures that I showed you earlier. And these then go on, if you maintain them in minimal media, to differentiate into kidney organoids with both tubules and podocytes. And these are some phase contrast images of what this process looks like over a couple of weeks, where you can see the emergence of these clusters of cells with tubules, these tubular structures, and each of these little tubular structures is what we refer to as an organoid. They're about 200 microns in diameter. And if you then go and stain these, we can see that there are nephron progenitor cells based on specific markers that are within these structures and are differentiating into kidney tubules. So how do we really know that we're looking at kidney versus any other organ? And I think the key to this is to compare the organoid in question, in this case the kidney, to actual human kidney tissues. And this is what's done in this slide. On the left we have a human developing kidney, and on the right we have the kidney organoid. And this also helps us gauge how well we've done in our recreation process. And all I've done here is stain these for a tubular marker, which is lotus lectin, and a podocyte marker, which is called podocalyxin. The tubules are in green, and the podocytes are in red. And you can see that they form separate clusters. The podocytes in the organoids actually form these really tight aggregates of epithelial cells, and they tend to be on the periphery of the organoid. And these are then connected directly to proximal tubular cells, which are in green. And these same types of arrangements are observed in the human kidney on the left, where you have the podocytes forming the filtering layer of the glomerulus, and this drains into these green tubular structures. And in both cases, these then continue to connect to more distal segments, which I'll describe momentarily. Now, I want to point out that the organoid and the actual organ are different from each other. They're clearly not the same types of structures, and particularly what we observe is that there's no vascularization in the organoid. So these structures don't have these really intense capillary loops that invade the podocytes and form the glomeruli. It's also less organized in general compared to the actual kidney tissue. So we still have a ways to go. But nevertheless, it's impressive to think that this structure here on the right, which at a rudimentary level resembles the kidney, actually was once a skin cell and has now totally changed its fate and has turned into many different types of kidney cell. If we look a little bit more closely at these structures and compare them to the structure of the nephron, we can see that the kidney organoids contain segments that very much resemble the principal segments of the nephron. And these are all epithelial cells, but they form these connecting segments, and these are the podocytes, which are the filtering cells, the proximal tubules, which is are the cells that the filtrate immediately is drained into, and then further down there are distal tubules, and these express different types of genes compared to the proximal tubules, even though it's all one connected segment. So this is quite remarkable, I think, that just starting with cells in a dish, they can somehow self-organize into these segments in just the right order, very similar to what we see in this highly complex nephron subunit in vivo. On top of that, we've also checked for endothelial cells, which are the blood vessel cells in these organoids. And we've observed that these are also present in the organoids. And this is some staining here on the right, where you can see the endothelial cells in red glomming on to a variety of these organoid structures. So the endothelial cells, they form chains, and they do interact directly with the podocytes and with the tubule cells, just like they do in the nephron. What they don't do in this situation is form actual blood vessels, but I think that it's still very interesting that these form at all and that they seem to be attracted to the organoids. And this isn't just an occasional observation in one field. It's actually something we see in 80–90% of all organoids that have proximal tubules. They also have podocytes and they have endothelial cells associated with them. And importantly, this isn't just something that our own lab has observed. It's actually been reported by several other labs that have made organoids in slightly different protocols. And for example, this is one here is listed below, the Takasato et al. Paper, which was published in Nature around the same time as our Nature Communications paper. And in all of these cases, I think the fundamental segmented structure of these kidney organoids is very similar. So now we've introduced the kidney organoid, and I'd like for the bulk of the talk to talk about how we can actually recreate kidney disease in a dish with these organoids at a level that we haven't been able to do before, and critically using CRISPR gene editing, which has been key to this process. So the importance of doing this, in my mind, going back to that Richard Feynman quote, is that we really don't understand how the different types of kidney disease actually work. In other words, at the molecular level, we don't quite understand why things go wrong and cause defects at the tissue level. And this is an example on the left of a very common kidney disease called polycystic kidney disease, where you can see a healthy kidney compared to a polycystic kidney. And what's happened in the PKD kidney is that the tubules, for reasons that we don't quite understand, have ballooned out and formed big sacs of fluid called cysts. And these eventually crowd out the healthy kidney tissue. And that's just one example. There are many, many diseases affecting different parts of the nephron that make up the entire spectrum of chronic kidney disease. And if you look at that spectrum, we really have very few medications that treat these specific disorders, that treat the root causes of kidney disease. And as a result, we're limited to these late-stage interventions like transplant or dialysis. So wouldn't it be great if we could treat the root causes of disease and actually prevent that kidney from becoming polycystic? Just to give you a sense of the magnitude of this problem, I've shown here a picture from a Coldplay concert that I attended earlier this year. And this is about half of CenturyLink field here in Seattle. So it's about 30,000 people. And I've done the math and calculated that the number of people shown in this image is approximately 1/400th of the world's polycystic kidney disease population. So this is a very common problem. And this is just one, maybe accounting for about 10% of all kidney disease. So you can imagine how many people are affected worldwide. Now, just to give you a little more detail about PKD, in addition to the kidneys forming cysts, we also see cysts in the liver and the pancreas. And there are also vascular aneurysms that occur, which resemble cysts in some ways. And the reason this is so common is because if one parent has the disease, there is a 50% chance of any of their children inheriting the disease. So as a result, it's propagated from parent to child, and it tends to run in families. Now, we know the genes that cause this disease, and these are called PKD1 and PKD2. A mutation in either of these genes causes the disease. And these are shown here in this cartoon. The gene products are called polycystin 1, or PC1, and polycystin 2, or PC2. So PC1 encodes a long transmembrane receptor, and PC2 encodes a channel-like protein. In addition, there's another receptor protein called fibrocystin. This is associated with a more rare form of PKD called autosomal recessive PKD, which affects children. So what do we know about these proteins? We know that they form a channel receptor complex at primary cilia. And cilia are these antenna-like organelles that jut out from the apical surface of tubular epithelial cells. And this is diagrammed here in this cross-section of a kidney tubule. And they need to form this complex at the cilium in order to prevent PKD. And if they aren't forming that complex properly, the result tends to be proliferation or apoptosis defects, fluid secretion, possibly losses of polarity, and this results in the formation of cysts all along the tubular segments of the nephron, ranging from the proximal tubules all the way down to the collecting ducts where the urine goes through. But we really don't understand at the molecular level what these genes or what these proteins are actually doing to prevent cysts in healthy patients. And we don't have good models for this disease because the mouse, although it can generate PKD-like disease, is not exactly the same as the human. And we really do need better laboratory models to understand human pathogenesis. So a number of years ago, we started attacking this problem by generating iPS cells from PKD patients. And we generated a nice cohort from 3 patients with autosomal dominant PKD, which is the most common kind. And these all had mutations in PKD1. And we also generated a couple of cell lines from the more rare autosomal recessive PKD. And we could stain these for a panel of markers which we associate with pluripotent stem cells and embryonic stem cells. And we showed that these are good iPS cells, and they also had normal karyotypes. Now, at the time, we didn't have any way to make kidney organoids, but we still wanted to know if these cell lines could teach us anything about the disease. So we said, okay, iPS cells themselves are a type of epithelial cell. They're not so different from the kidney epithelial cells. Maybe they have cilia. Maybe they express polycystin genes. So here on the left is a Western blot where we're looking at polycystin 1 expression in embryonic stem cells compared to a couple of positive controls. These are mouse kidney cells, and the middle lane has been transfected with polycystin 1. And what you can appreciate is that the ES cells actually express a significant amount of endogenous polycystin 1 and polycystin 2, which are the genes affected by PKD. This is still in the undifferentiated state. So we asked, do they have cilia? And indeed, we showed that pluripotent stem cells, whether they're ES cells or iPS cells, do make primary cilia similar to kidney tubular cells. So now we asked, were the polycystins properly localizing to the cilium in our mutant lines compared to controls? And what we observed is that whereas in the controls, and as well as in the ARPKD patients, we saw nice co-localization of polycystin 2 with acetylated tubulin, which marks the cilium. So this was in about 50% of all the cilia had polycystin 2. In the PKD patients, the AD PKD patients who had mutations in polycystin 1, we saw a dramatic reduction in the number of cilia that had polycystin 2. And this was an interesting and exciting result to us. And for the first time, this really was giving us insight into the potential of pluripotent stem cells for studying some aspect of polycystic kidney disease. And we were seeing this in all 3 of the patients, suggesting that it was a real phenomenon. And it was teaching us that polycystin 1 was actually important for driving polycystin 2 to the cilium, that there was a direct relationship between these 2 proteins. And that at the time was still rather controversial. So as a next step, we obviously wanted to generate kidney organoids from these iPS lines and see whether they could also reveal or recapitulate features of polycystic kidney disease. And the good news is that we were able to generate kidney organoids from our autosomal dominant PKD iPS cells and our autosomal recessive iPS cells, as well as control iPS cells and embryonic stem cells. In all cases, using one protocol or the other, we were able to generate kidney organoids. But the bad news is that there was a lot of variability in the efficiency of generating these structures from one iPS line to the next. So here is a chart that we made summarizing, using a couple of different protocols, our ability to generate kidney organoids quantitatively from any given iPS cell line. And what you can see is that it's all over the map. Some lines respond well to protocol A, other lines respond well to protocol B, and there's really no rhyme or reason to this. It didn't seem that there was any specific defect in a PKD line versus a non-PKD line. It was rather more of a general variability that we were observing over the 21-day time course of generating these organoids. So this teaches us that every iPS cell line is different in its ability to differentiate, and I think this is a general phenomenon in the field. And the reason for this is, I think, that humans are very diverse. Our genetics are very diverse. You know, each of us has many different mutations compared to our neighbor. And these may have nothing to do with the specific gene in question that we're interested in studying. It has something to do with the other 20,000 plus genes in our genome. On top of that, we have to recognize that the mutations that occur naturally in the human population have certain limitations, and they're frequently rather mild compared to the sort of knockout mutations that you might hope to get a phenotype from. So as a result of this, for example, most patients with PKD only get phenotypes, they only have clinical symptoms around middle age. So it takes about 50 years for the disease to develop. And obviously, when we're working in a petri dish, we don't have 50 years to sit around and do our experiment. So we needed some way of overcoming these variabilities and limitations of naturally occurring mutations. So around the time that we were doing these experiments, CRISPR-Cas9 gene editing essentially emerged as a means of introducing mutations into the genome. Really the heart of this is Cas9, which is a programmable nuclease that searches DNA for short sequences. And this is sort of this blob here, this yellow blob represents Cas9, and it associates with this guide RNA that essentially gives it the directions where to go in the genome. And following these directions, Cas9 can associate with double-stranded DNA. It can unravel it and introduce a double-stranded break selectively at the site that it's targeted to by the guide RNA. And this can be used to then either introduce random mutations through non-homologous end joining process that's shown on the left here, or it can be used to introduce specific types of corrections as shown on the right using homology-directed repair pathway. So you can think in a simple way of these of this technology is kind of like a word search function and a word processor, whereas you type in more letters here into our search box, it progressively isolates a specific sequence within the genome. And what this allows us to do is perform gene editing with high specificity and high efficiency. So we get around 5% efficiency in our pluripotent stem cells. And it's also rather rapid, it's not expensive, and because it's using Watson-Crick base pairing, it really does speak the language of molecular biologists. And as a result, CRISPR has really caught on for many different types of applications throughout the molecular biology world. So I just wanted to run through our general protocol for generating mutants with CRISPR. And what we do is we use two plasmids. One is a Cas9 tagged to a GFP, and the other encodes a guide RNA. We transfect these into human pluripotent stem cells, again, in M-teaser under essentially maintenance conditions. And a proportion of these stem cells do take up the Cas9 GFP, and we can then isolate those by fluorescence-activated cell sorting. We then plate these out, and they form individual colonies, and we pick the colonies and expand them. Half of them we freeze, and the other half we submit for screening by PCR to identify the colonies with mutations. And these are just some images that we've taken at the flow sorting step of this process. So here on the lower right, you can see the expression of the Cas9 GFP. This is our gating that we use to select the pluripotent stem cells. And then after replating, it's always useful to go back and make sure that the pluripotent stem cells are indeed expressing the Cas9 GFP. Now, this is a transient transfection, which means that the Cas9 GFP is only on for a short amount of time. After that, it turns off, and the mutations that are made are then permanent in the pluripotent stem cells themselves. So using this approach, we were able to generate pluripotent stem cells with mutations in either PKD1 or PKD2. And these are examples of some of the chromatograms that we were able to obtain, where you can see right at the spot that this guide RNA is targeting, where that arrow is, suddenly the chromatogram sequences become distorted. And that's a sign that you have multiple sequences there. And then you can go back and make sure that they actually have indel mutations, insertions, or deletions that would disrupt the gene. Now, even if you have nice DNA sequences, it's always advisable to go back and perform protein expression analysis by Western blotting. So here we went back to our tools that we developed, where we were able to perform Western blots for polycystin 1 and polycystin 2 protein. And we can show that in our PKD1 knockout mutants, there is no mature isoform of PC1. And in our PKD2 mutants, likewise, there's no polycystin 2 being expressed. So now we have paired mutants and controls of the same genetic background, and the only difference is the single mutation in PKD1 or in PKD2. And these are severe mutations, more severe than what we would normally find in a human patient, which I think increases the likelihood of us being able to see an effect. And I'll point out that the stem cells that we generated using this process didn't show any general problems. In other words, they divided normally, they seemed healthy, they formed teratomas in animals, so they could differentiate very well, et cetera, et cetera. So the first thing we really wanted to do was go back and take a look at the levels of polycystin 2 at the cilium in these new CRISPR mutant stem cell lines. And this was important because before, when we were doing it with iPS cells, we did see some variability from patient to patient. So we wanted to make sure that our previous conclusions, that PC2 depended on PC1, for ciliary localization was actually correct. And so here are some images showing polycystin 2 expression co-localized with cilia, and you can see in the controls these undifferentiated stem cells have PC2 at the cilium, whereas in the PKD2 knockout, there's really no PC2 staining. And what was nice to see is that also in the PKD1 knockout, PC2 was not observed at the primary cilium. And this was a very dramatic loss of PC2. It was essentially 100% loss of PC2 from the cilium compared to the controls. So this validated our previous finding that PC2 was dependent on PC1 to go to the cilium. Now we went and said, okay, what happens if we make organoids? And you'll remember that when we used our iPS cells, there was a lot of variability in the differentiation from iPS cell line to iPS cell line from different patients. So now when we do this with the same genetic background, we see much more similarity between the wild type, the non-mutant pluripotent stem cell lines compared to the PKD mutants. So I think we've successfully overcome a lot of the noise that was in that system. And now we can compare these as apples to apples rather than apples to oranges. So when we initially differentiated these, we didn't see much of a difference between the mutants and the isogenic controls. They both formed organoids pretty nicely, so we just waited. The protocol, the organoids are differentiated by about day 21, and we gave them another couple of weeks. And what we observed is that over that period of time, we saw structures like this one shown here in the phase contrast, which is sort of shaking a little bit in the culture media. And this looked like a balloon-like structure, a hollow structure. And indeed, we could stain it for lotus lectin, which is an epithelial marker of the proximal tubule. And we could see that this was essentially a hollow structure, which was lined by an outer layer of epithelial cells. And at the very bottom of this structure here, you can see the remnants of an organoid from which this cyst has emerged. So this was a very interesting finding. And when we quantified these, we observed that they were only forming in our PKD1 or PKD2 mutant lines, whereas the isogenic organoid controls didn't form cysts. So that meant that this cyst formation was really a specific phenomenon to PKD mutants and was being caused by these mutations. And importantly, this was happening in both of these genes. So it wasn't just some sort of off-target effect of one of our guide RNAs, because if that were true, it wouldn't happen to both the PKD1 and PKD2 guide RNAs. And instead, it was occurring specifically to the genes in this PKD pathway, which is so poorly understood. Now, one limitation of this finding is that it was only true in about 10% of all the organoids in the dish. So even though all the organoids had biallelic mutations in these genes, only a fraction of them actually went on to give cysts. But it was enough to be detected, and again, we had the negative controls there, which really established it in a clear way. So this was a very exciting discovery for us because we actually recapitulated at some level the phenotypic hallmark of PKD, which is the emergence of these cysts from kidney tubular structures. And what this teaches us, I think for the first time, is that PKD cystogenesis is actually a cell-intrinsic process that we can reconstitute in a petri dish in vitro. There's something about these genes that is so important to preventing cystogenesis that it can actually be boiled down to this very simple cellular system with defined ingredients. So we published that in the Nature Communications paper as an illustration of the power of kidney organoids to really recreate disease processes. And I think that all the previous studies that have been done really in PKD have never really been able to achieve this type of in vitro model due to the complexity of the disease. People have been able to make cysts, but the cysts have not been specific to the PKD mutations. So this, I think, was an important step in our understanding of the disease. So what we've done subsequently is use this system to really try to understand how these cysts are forming. And here we have a movie that we've taken. We're taking images every 4 hours, and you can actually see the differentiation of the tubules. And because this is a PKD organoid, you can see how this little cyst pops off of it on the left. And what we observed is that the formation of these cysts always occurred together with a kind of detachment from the underlying plate. And this was true in both PKD1 and PKD2 organoids. The formation of the cyst involved essentially a ballooning out of the tubular segment to form a precyst, and this detached from the plate and then subsequently expanded to form a rapid cyst. And what this suggested is that the process of cystogenesis actually involves detachment of tubules from the underlying substrate, that this might be an important step in the cyst formation process. So we then thought about this and we said, would we be able then to change the time course of cystogenesis by changing the attachment situation? And to test this, we came up with a new protocol where we differentiate the organoids in adherent cultures as we normally do for 21 days and allow them to form organoids, but these have not yet formed cysts. And then we take these organoids out and we place them in suspension culture for the rest of the 2 weeks to test whether detaching them manually would actually promote cystogenesis. And this is a little movie here where you can see how this works. We take literally just a needle tip and micro dissect out these little pieces of organoid and then aspirate them and place them into a suspension culture similar to what people use for embryoid bodies. And when we did this, we observed that in both PKD1 and PKD2 mutants, we very rapidly formed cysts from a high number of these organoids in suspension compared to the control situation. If we let these cysts stay in suspension for longer periods of time, they continued to grow, and we could get these very large structures shown here. So this is a 6-well plate. On the right, we have organoids with PKD mutations. On the left, we have the isogenic non-PKD organoids. These were differentiated side by side, placed in suspension, and grown for several months. And you can see that the gene-edited PKD organoids are forming cysts that are about 1 centimeter in diameter. So these are easily seen by eye. And this is an example. This is just a movie we took just the other day where you can see one of these 6-well plates, and we're sort of zooming in, and you can see the size of these cystic organoids after a few months in culture. When we quantify this, we see that over 80% or so of the organoids actually go on to form cysts under these conditions in the mutants, whereas the number of cysts in the controls remains very low. So we've now been able to really greatly increase the efficiency of organoid formation, of cyst formation from our organoids compared to our original adherent cultures. So this really does point to a critical role for attachment or microenvironment in PKD. When we analyze these for cell division events, we find that they are actively growing. There's many dividing cells in the organoid. This is a cyst that we're kind of looking at in 3-dimensional view, the lower portion of a cyst. We can count the number of mitotic nuclei, and we see that it's increased in the PKD cysts versus normal kidney organoid tubules. And by performing microarray analysis together with the lab of York Pei, we've been able to observe that growth pathways and proliferation pathways such as mTOR and c-Myc are upregulated in these cysts relative to the organoids that they're coming from. So this is consistent with what we know about polycystic kidney disease being a proliferative disorder. Now, to really understand these, we need to compare them to PKD cysts in vivo, and that's what we've done here. We can take our organoids and compare them by histology stains. And this is actually an autosomal recessive PKD patient, so it's a developmental disorder where they're forming cysts as soon as the kidneys are forming. And we found that the appearance of this epithelium in the cysts in our organoids is actually pretty similar to the appearance of the epithelium in these PKD patients. And we furthermore can stain these and look at the different nephron segments that I talked about earlier to see which are actually giving rise to the organoid cysts. And we find that the organoid cysts are coming from both proximal and distal tubular cells based on specific stains and not from podocytes. And this is very similar to what we see in patient cysts in vivo. And here quantified, you can see that almost all cysts have both distal and proximal tubular cells. In addition, we see that these are indeed epithelial cells lining the cyst, so they express the tight junction marker ZL1, and they have primary cilia, which is this antenna-like organelle, which is fundamental to the disease. So we can actually go back now and compare our disease model to our actual disease tissues to see how well we've recreated that disease. Because our findings were pointing to a role for microenvironment, we also tested the effects of planting organoids in a solid microenvironment. So we took individual organoids and placed them in these large balls, these droplets of collagen. These are about 3 millimeters in diameter, and we asked what would happen both in the PKD and the control. And interestingly, over a couple of weeks, these tiny little organoids actually surrounded these droplets and compacted them to a much smaller size, as shown here. So you can see that the organoid here is only 200 microns. You couldn't even see it in this image, and yet it has expanded and contracted down this big piece of collagen. And interestingly, the PKD organoids were deficient in their ability to actually change this collagen droplet. And they could still compact it, but it wasn't to the same extent as the control organoids. And these are images showing the geometry of this collagen droplet after it's been compacted. Essentially, it's still fully collagen on the inside. And what's happened is that the epithelial cells in the organoid have wrapped around this droplet and crushed it down to a much smaller size. And here you have serious red staining showing the collagen content. And you can see it's much denser and smaller in the control versus the PKD. So this again points towards a role for attachment and microenvironmental interaction in the way that these proteins are actually functioning. So we wanted to know what the molecular player might be that's involved in this microenvironment interaction. And the obvious candidate really was polycystin 1 because it looks like a receptor protein. So we examined the levels of polycystin 1 in our organoids in control and in our polycystin 2 mutants. So now we're looking at the effect of polycystin 2 on polycystin 1. And what we observed is that there was a great reduction in the expression of polycystin 1 in our PC2 mutants compared to the control. Even though there were no mutations in that gene, it seems that the PC1 protein was actually required to stabilize, or rather the PC2 protein was required to stabilize PC1 expression. And we saw this in both the organoids and in undifferentiated pluripotent stem cells, indicating that this was a general phenomenon. And actually, you can see the loss of PC1 in this cell line, this PC2 mutant cell line, is as extensive as the loss of PC1 in the PKD1 mutant cell line. And that was kind of surprising because mouse studies had suggested that while PC2 may be important in maturing PC1, it wasn't really that important for its expression. And again, what is polycystin 1? It's essentially an adhesion. It looks like an adhesion-coupled receptor of some kind. So it has a really long transmembrane domain with many different subdomains. And it looks like it's binding to something on the outside of the cell. But we don't yet know what that ligand is. So this is a fundamental question. Even after 20 years of knowing this gene, we still don't understand what it's binding to. And our guess would be that it's playing a role in adhesion to the microenvironment. So what all these studies really give us is a new model of organoid PKD cystogenesis. And we started off with our adherent culture system where you have organoids, and they're essentially in an equilibrium where most of the organoids stay compact, the tubules stay coiled, and a small minority of these organoids in the PKD mutants are lifting off from the substrate and forming cysts. Now, when we instead remove the organoids and place them in a suspension culture condition, what this does is it removes those adherent cues that were dominating the structures in the earlier culture. And this unveils the phenotype of PKD cystogenesis, wherein the mutant organoids can very rapidly unspool and expand out to form a cyst. We don't yet understand fully how this works, but I think it really does point to a critical role for microenvironment in polycystic kidney disease. And this is a role that has been relatively overlooked in the past decade as the focus has been on cilia and other parts of PKD function. And we published a nice paper about this in Nature Materials last year, which has all these details and more. So in the final part of the talk, I would just like to discuss the future directions and conclusions of the work. And I'd like to just return to our model, our paradigm here, where we're generating kidney organoids from patients with the long-term goal of transplantation. And as we think about gene editing in this context, we might say that if a patient had, let's say, a genetic mutation, we may have an opportunity at the iPS cell stage to change that genetic mutation or correct it before we make a kidney organoid. And this remains a frontier for us to take patient cells and be able to correct their genetic mutations and compare them to the original cells. And ultimately, this would provide a pathway where we could be able to, if we were to put these tissues back into the patient, we would be able to implant them as gene-corrected grafts, so they wouldn't then re-suffer the effects of the disease. In work that I haven't had time to discuss today, we've also used CRISPR in kidney organoids to model defects and diseases of the podocytes, which are the filtering cells of the kidney. So this is a model of nephrotic syndrome. It's actually teaching us a lot about how these very specialized epithelial cells differentiate and form the glomerular filtration barrier to begin with. And I think this work, together with our work on PKD, really does suggest that we can use this system to create remarkable changes in the kidney organoids, and that it will be a good system for studying human disease mechanisms, particularly as they relate to early development, which is really the stage that the organoids are at. A frontier for this work is putting these organoids into higher throughput formats where we can perform clinical trials in a dish, so to speak, and screen hundreds of thousands of drugs or compounds for their effects on organoids. So this is a very powerful type of system which has previously really been limited to very simple monolayer cultures. And now we can contemplate doing the same type of experiments with our very complex organoids. And this is an example here on the right of the robots that we have that we can now use instead of generating the kidney organoids by hand in larger wells. We can use these robots to generate them on these tiny plates and then perform thousands of experiments in parallel. And I think this is going to be a very useful technology, a new type of big data experiment that will produce functional data for us to analyze and possibly figure out how these diseases are working, what pathways are involved, and what types of drugs we could potentially use to intervene with them. Just as an example of that, we've been looking at different drugs that are believed to be involved in pathways affecting PKD. This is one of them. It's called forskolin, and forskolin is known to activate cyclic AMP signaling, and cyclic AMP upregulation is believed to be one of the ways that PKD works, although we don't yet fully know how that pathway might operate. So to test this, we actually added in forskolin to both control and PKD organoids, as shown here, and we took time-lapse movies. And what you can hopefully appreciate is that in both the control and in the PKD organoids, addition of forskolin results in the rapid formation of the cysts in these cultures so that the tubules rapidly swell up in response to the cyclic AMP activation. But what's interesting here is that this doesn't seem to be any worse in the PKD cultures versus these controls of the same genetic background. And that implies to us that although cyclic AMP activation can indeed cause this to form, it doesn't seem that the PKD organoids are sensitized to it at a higher level than the controls. And therefore, this may be more of a modifying pathway than the direct pathway by which the polycystins operate. We still need to do more experiments in this system to really investigate this, but it is important because right now, the only drug on the market to treat PKD actually, it targets this specific pathway. Finally, I'd like to just spend one slide talking about the potential of the organoids for kidney regeneration in vivo. And these are some of these experiments that we performed. This was an early experiment from the Nature Communications paper where we actually took organoid cultures that were differentiating them, and we injected these differentiating cultures into the kidneys of newborn mice. And then we stitched the mice back up. They're totally fine, and they grow for several weeks with these human grafts. These are immunodeficient mice, so they can tolerate and actually are fine with human cells in their bodies. And when we did this, we found that there were human proximal tubules that were forming within the kidneys of these animals. And this is a picture here showing a mouse proximal tubule, which isn't stained with red because it's of mouse origin, right next to a human proximal tubule in the same tissue that is stained with red because it's of human origin. And that was exciting because you can see the proximal tubular marker is actually pretty similar in the mouse and the human tissue that's grown up together. And more recently, we've been doing experiments in adult animals, and these are shown here. We've injected beneath the kidney capsule of mice kidney organoids, and we've allowed these to differentiate, as you can see here on the right. And what's interesting here is that you can see the graft is on the lower part of the image. The mouse kidney tissue is actually on the upper part of the image. And there's tubules in the graft. These are labeled with the tube here, compared to the mouse tubules, but they don't really look very similar. The human tubules here look kind of wimpy compared to the mouse kidney tubules. That's the first observation. The second observation is there's a lot of other types of cells filling up around the tubules in the human graft, and there's also a big lump of what appears to be cartilage forming in this graft. So in general, I would say that we can get kidney organoid grafts to survive in a mouse subcapsular kidney transplant, but the grafts probably have other types of cells besides kidney coming along with them that could be dangerous. And the grafts at this point are not making mature tissue that looks really anything like the nice kidney tissue of the mouse. So I would be very scared to implant anything like this into a person. I think we still have a long way to go before we'll be able to make functional grafts in animal models and certainly in patients. So in the short term, I think the focus should be on the disease modeling aspects of these organoids. So with that, I'll just give a few conclusions. I've shown today that we can generate kidney organoids with complex nephron-like structures from human pluripotent stem cells. This is the first time we've been able to make these types of structures. We've used gene editing and been able to recapitulate the phenotypic hallmarks of polycystic kidney disease in organoids. This includes loss of polycystin 2 from the cilia, but really most impressively, the formation of cysts from kidney tubules. Moreover, by toggling 3D culture conditions, we've been able to reveal a critical role for microenvironment in polycystic kidney disease that was really, I think, underappreciated previously. And this shows that we can use this system to really learn new things about our disease in question. And finally, looking forward, we think that organoids together with CRISPR technology really raise new possibilities for both drug discovery as well as cell/regenerative medicine. So with that, I'd like to thank all the people who are involved in this work here at the University of Washington, particularly Nellie Cruz, who really did the yeoman's share of the work with the PKD project, as well as everybody else in our lab and our close collaborators. I'd also like to thank our mentors and collaborators at Harvard, where the work was originally begun. And I'd like to thank our funding organizations, in particular the PKD Foundation and NIH, as well as the ASN and the National Kidney Foundation, and our divisions and departments here at the UW for supporting this work. And I will thank you guys for your attention and take any questions.
Okay, thank you very much, Benjamin. That concludes the presentation segment of the webinar. We will now begin our Q&A session by addressing the written questions that were submitted throughout the presentation. So let's start with our first question. How important do you think vascularization will be for your organoid model in the future, and do you think this will be achievable?
I think that vascularization is a really important part of the kidney. And as we, it depends what our application really is. If we're interested in a disease process that requires vascularization, then I think we'll need to vascularize. And there are many disease processes, particularly those affecting the glomerulus, that really do involve the interaction between the blood supply and the podocytes, for example. And there are also vascular diseases that affect, the tubular portion of the organoid. So I think that for vascular diseases, there's no question it will be very important. In terms of the achievability, and for regeneration, I think it's also likely to be highly important, obviously. We're not going to get very far by implanting structures that can't become vascularized. I'd say that, we don't yet know how easy this is going to be to do. I would argue that, if you look at in vivo transplants, there's been examples of some kind of vascularization. In other words, the grafts of almost any kind that you get to grow in an animal will involve invasion of the graft by the host mouse endothelial cells, and this is how the graft gets its nutrients. So there is some potential for vascularization, but is it going to be able to form a real glomerulus-like structure? That I'm less convinced. And I think we should be careful to differentiate between bona fide glomerulus formation involving capillary structures and filtration compared to sort of some blood vessels here and there in a disorganized way. Now, if you look in vitro, I would say that there's really no example of anything really becoming vascularized in a bona fide way in any system. So in vitro, I think that's a major challenge to get things to really become vascularized, where they're not getting nourished from the media surrounding the cells, but rather through a blood supply. And there again, I think it's important to really differentiate between things like what we're seeing in our dish, where you get some endothelial cells stuck in among the cultures, versus a true vascularization event. So I think it's going to be harder to do in vitro, maybe possible in vivo under the right conditions. And in general, I do think it's going to be important.
Okay, thank you, Benjamin. Second question, are the pictures that you presented from earlier in the presentation whole mount staining or from sections of the organoid?
So one advantage of the way that we do this is we just grow them in adherent cultures, and this spreads out the organoids so that they're not in like a giant pellet. So these are all images of whole mounts. Essentially, these are just grown in the dish. We fix them in the dish, and we stain them right there in the dish, and we don't have to do any sectioning. So that was for the earlier pictures. Some of the cysts we did do bisection, just because we wanted to be able to accurately compare them to the tissues in vivo, which were prepared by paraffin sections. But for the vast majority of the images that we show are just straight up confocal images of structures that were stained without any sectioning.
Okay. Which marker did you use for the endothelial network?
So the endothelial network that we were showing there was with CD31. It's a antibody. It's a mouse antibody from BD that recognizes human CD31 pretty well. We've also used other types of markers more recently that confirm that these are indeed endothelial cells.
Okay, next question. It is thought that endothelial cells migrate to the embryonic kidney from surrounding tissue. How would you interpret where the endothelial cells in organoids come or differentiate from? Do they differentiate in response to chiron or MPS niche signals?
Well, I think that if you look at how endothelial cells differentiate in other types of pluripotent stem cell cultures, they do show up in response to chiron. So you can definitely induce them that way. I wouldn't be surprised if that was playing a role here. We don't see a lot of them in our cultures, so obviously this could be further optimized to probably differentiate more of them. I don't know if you didn't have the chiron, whether you'd also see endothelial cells in these cultures or not. So far, the endothelial cells have been seen, I think, in the literature only in protocols that have used the chiron. And we don't know whether these are really kidney-specific endothelial cells or just endothelial cells that are coming from other places in the body. We're sure that these organoid cultures are not 100% pure or full of kidney. There's a lot of kidney in there, but there's also a lot of other lineages.
Following genome editing, did you test for any off-target effects? And if so, what strategy did you use?
So It's very difficult to really test for off-target effects because you'd have to sort of interrogate the entire genome. The strategy that we've used to address this question in general is to not do any of our experiments with a single line or a single gRNA. I think this is the general strategy that's been adopted by the field. So in our case, we've used both PKD1 and PKD2 directed mutations. We see the same phenotype in both. So that's very unlikely to be an off-target effect. In addition to that, for each of these genes, we do the experiments with multiple cell lines. And that's important because actually there is some variability even among isogenic clones from one cell line to the next. And we don't know why. But because that variability exists, you got to do your experiments in at least 3 clones of each different genotype to really make sure that whatever effect you're seeing is real. And similarly, we do the experiments with multiple control lines. And I should point out that we've also made mutants in other genes like podocalyxin, which is the one that we use to study the podocyte, and there we don't see cyst formation. So we know that just the process of CRISPR is not somehow inducing a selection event which promotes the formation of cysts. Nevertheless, I think this is a very important question. It's really critical that when doing these types of experiments, we get the controls right.
Thank you. And which method, either non-homologous end joining or homology-directed repair, was used to generate the PKD organoids? And would the choicing method have any consequences for cystogenesis?
That's a good question. So the only method we've used is the non-homologous end joining method. And that's the dominant pathway that comes up when you use CRISPR. It's harder to do homology-directed repair, but the advantage of that is that you can introduce very specific mutations. That's definitely a frontier for us. And what you might be able to do with homology-directed repair is, pinpoint, I think, the effects of very specific mutations like point mutations and things like that, as opposed to just knocking out the genes. So that's an interesting frontier for the future.
Did you stain the organoids for the presence of primary cilia?
Yes, we have, and I showed a little bit of that data, and it's also in the Nature Materials paper. So we do see primary cilia in the organoids, and certainly in the cysts. There's many primary cilia in these epithelia. And in general, these tend to be on the apical surface, right where you would think they should be. We don't yet know, though, whether these primary cilia are as good as what you would normally see in humans in vivo, and we have to do more work, I think, to really characterize these primary cilia.
Thank you. And are you looking into any other functional tests for the organoids other than cisplatin or gentamicin assay and dextran uptake?
Are we looking into any other functional? Well, the disease models themselves are a functional test. They're really a better functional test, I think, than nephrotoxins, which are But I think the nephrotoxins are also very interesting in their own right. There's all sorts of things that kidneys do. They're very complicated. So they reabsorb, they secrete. The podocytes can respond to certain types of stimuli. I mean, there's a world out there that one can look at in terms of function. And I would say we're trying to look at some of these functions, but there's no way any individual lab is going to be able to look at all of them. So I think we're really kind of focused right now on some of the perhaps more classic ones, and the other ones I think are out there for labs with specialties in those areas to really chase down.
Okay, so I think that's all we have time for today. I really want to thank everyone for attending, and I'd especially like to thank you, Benjamin, for your time and an excellent presentation. Thank you very much again, and goodbye.